I feel that the pharmacogenetic has a huge role and important role in future, and it can basically balance between the desired and undesired effects of the medication according to genetic variability of each patient. With such a revolution in genetics and fast-paced growing of genetics and IVF, I feel that this is going to be our future. I feel that we need more research to be done, and knowing more about the physiology and different pathways, and also pharmaceutical companies have to start to think out of the box and investigate different form of the FSH, because it's always the matter of the ligand and receptor. So all the studies are done on the receptor, but not so much on the ligand, and they are all producing the same FSH, purified FSH all the time. So I think there is a huge need for more research from the pharmaceutical company as well as the academic and research groups to know better. So the I thought to talk about the pharmacogenomic in ovarian stimulation, and it is a very, very new area of science in IVF. There are not so much data exists, but I try to go through and summarize everything to make it work. So I don't have any conflicts of interest. So from the time that Louis Brown were born, it takes about 40 years now, that the demand for the IVF has been increased dramatically, but the success rate, the protocols, and medications are still the same. Still, 60 to 50% of the stimulated cycle end up to failure of pregnancy. So it's important in this way that to consider some way to personalize and tailor the treatment according to the patient's characteristics through the genetic variability in patients. So what is the pharmacogenetics or pharmacogenomics? It's a study of the genetic variation in response to the drug treatments. It's a very important example form of the precision medicine. And according to the variability, genetic variability, basically it can has some effect and improvement on balance between the side effects and desired effects of the medication. From the history standpoint, it's basically started from 500 BC. That was the first time they find out that some people, when they get the fava beans, they will develop very severe side effects, and they will, some of them die. Then around 18th century, then the hereditary form of the disease and pattern were found out at that point. And the name of pharmacogenomic started to go around 1957. And in the 20th century, basically the huge improvement and breakthrough findings has happened, like G6PD, and also finding of the some association of some genetic abnormality with the patient's characteristic or the disease. The huge revolution was in 2000 to 2003 that they sequenced the human genome. And from that, everything has changed in a way that now we have more information with regards to association between our genome, how the physiology works, how the medications work, and why we have so much variability. The JBOS studies are the cornerstone of pharmacogenomic studies. So how does it work is we divide the patients, one group with the specific phenotype, and compare them to the control. First, these patients, they do the sequencing and go for the SNPs, and then they will find out that some of the SNPs are more common. They can see statistically significant in some region. Then they can sequence the region to find the candidate gene, and if the pathogenic variant is more than 1%, then they call it polymorphism. Is less than, they call as a mutation. The The caveat is here, though. In the human disease, J was the effect size is small or it's moderate. And that makes it not very valuable in basically understanding of the genetic of the hereditary or the disease, because there's so many characteristics of the patient, environmental factor that should be in consideration. However, the difference is in pharmacogenomic, because it's dichotomous basically variable. So it's yes or no. And by that way, the power and effect size in JWAS study with regards to the pharmacogenomics studies are pretty good. So what are the applications of pharmacogenomic? It can improve the drug safety, can reduce the adverse effect, can tailor the treatment to meet the patient's genetic predisposition and find out what is the optimal dose for this patient. It can improve drug discovery and also improve the proof of principle for efficacy studies. So, how does the pharmacogenomic works? There are good association between the pharmacogenomic, pharmacodynamic, and pharmacokinetic. The genetic variability in each person can determine what is the metabolism, distribution, or absorption of the drug. For example, if the cell has more receptors, then the response is more. If they have less receptor, the response is less. Or the other things is drug breakdown. If the enzyme metabolized faster, and they're fast metabolizer, then you need to give the medication either more frequently or you have to increase the dose. And if they are the slow metabolizer, then definitely you have to decrease the dose of medication in the patients. And also drug uptake has another effect on pharmacokinetic. How many receptor can go and uptake by the cells. If, for example, this number is low, means that lower amount of the medication goes inside the cells, so we are going to see the adverse effect of the medication according to the genetic variability and how the gene express of the receptors and enzyme activity on tissue. Now, with regards to the controlled ovarian hyperstimulation, I'm sure all of you saw many patients that ovarian reserve is very good. They go through stimulation, very unexpectedly very poor response. Or you have somebody who doesn't have a very good AMH or AFC, but they go through stimulation and unexpectedly makes us happy and we get so many eggs. Why this happens? Why some people develop OHSS and some people don't? And honestly, I have seen even with the estrogen of 3,000 or 2,500 OHSS, which shouldn't happen. So these are all the questions that could be responded and answered by study on the genetic variability on human and its association with the controlled ovarian hyperstimulation. And also it can help us to tailor the medication for each patient. This is the slide that shows the most common polymorphism that has been studied in association with the COH or outcome in IVF treatment. I'm not going to go through all of them. The main one is the FSHR polymorphism. And receptor of FSH, as you know, it's one of them that has been studied most than any other basically polymorphism. And the physiological action of the FSH really depends on activation of the receptor, and usually this expressed by granulosa cells. Now, there are, between all of the studies, there are two variation or polymorphism that constantly, when they studied, hit the target and showed that this polymorphism are associated with the different response or variability. One of them is the position of 307 alanine to threonine, and another one is the aspergine 680 to serine. And both of this polymorphism in patients were associated with the lower response to the treatment. However, if we have for example, a position of 68, we have serine 68 serine, then it will cause OHSS, and the studies shown that these are the patients could be used as a marker to predict who's going to develop the OHSS. Also, not only that, the 307 threonine to alanine has been shown that not only the duration of the stimulation is longer, the amount of the FSH that this patient needs is higher, the amount of the matured egg in these people are lower, fertilization lower, and also the blast formation are lower. And basically, these two polymorphisms, when they are together, then even the pregnancy rate will be dropped. Gain of function mutation usually are the one that makes the, for example, an FSH receptor to work more, right? These are majority of the time happens in the extracellular domain of the receptor. And when this happens, usually they work like a autosomal dominant. However, the poor responders or inactivating or loss of function polymorphism, usually they happen in through all the three parts of the receptor, and therefore they have to be the they are autosomal recessive, means that you have to have two different genetic variants or compound heterozygous to have the phenotype. This slide shows that the most actionable and functional pharmacogenomic variants in FSHR gene. Majority of the deletions and loss of functions happens in the exon number 7 and exon number 10. Now, let's look at the different of the changes that could happen in the receptor. For example, if we have the polymorphism in the exon 2 splice side, in the splice side that can delete the exon 2 on the FSH receptor, then we are going to have the poor responder and lower response compared to the wild type. Another one is the leucine-rich region in FSH receptor that affects the extra domain, and therefore it will affect the binding of the ligand to the receptors and affect the response and therefore the availability of the medications. Now, if you look at the these two polymorphism, one is basically in the polymorphism in the region of the 6165 that has the basically substitution of the A to G in position of 919. And they find out that if we have a homozygote of AA in this group, they have much better response, this patient, compared to any other alternative, and they got the more retrieved oocyte during the stimulation. And another one is in 6166 in the close to the 3' region, and they find out that the GG homozygote of this patient had the higher level of FSH than the other forms. But when you look in the PCOS patient, but when you look at this polymorphism in non-PCOS patients, then it will show that the same polymorphism could cause a poor response. Also, another important one is the polymorphism that affects the 29 base before the exon number 1, and it has been shown that it's associated with the low functionality and gene expression. Basically, the amount of the receptor expression on the cell surface will be decreased in these people. Recently, a novel variant of FSHR receptor has been shown, and that's the one that basically changed the isoleucine in a position of 423 to threonine, and this affects the second transmembrane domain and affect the basically membrane expression of the FSHR. Now, the importance of this was that when you have this polymorphism, this affects selectively the CAMP pathways of the FSH receptor, and the two other ERK1 and 2 and arrestin less will be affected. So as you see, the genetic variability and these variations, although they are in infancy, but they can show us that these are going to be the future. Then we can personalize accordingly the dose of medication and also how to manipulate different pathways in order to get the better response in these patients. This all I talked was about receptor. Now let's talk about the ligand. Are they going to affect as well or not? I'm going to touch very quickly on natural FSH or human FSH. All of you know it has two alpha subunits and beta subunits. The alpha subunit has the 92 amino acid and also some sugars added to these parts. And beta subunit has 111 amino acid, and also it has some sugar parts. On the top of the sugar parts of the alpha and beta subunits, usually sialic acid will be added, that it will change the acidity and also the half-life and effectivity of the medications. Now, if we have the fully glycosylated FSH, they call it as FSH24, and this has usually difference in expression throughout the female reproductive life. The two other forms of the FSH, one FSH18 and the other is FSH21, they have two glycosylation. And then, so they have less acidic form. So these are also our different expression throughout the life. When we are younger, the amount of the ratio of 21 to 24 FSH is higher. As much as we go through the reproductive life, this ratio will change and FSH24 will be added. More glycosylated and more acidic, so different physiology and different action. Means that I said that means that when we are in menopause, usually we have highly acidic human FSH. The FSH of 21 and 18 diglycosylated, they are much stronger in attachment to the receptor and activation of the receptors. Now, this slide, don't be scared, I'm not going to go through all of it. It's just showing that what are the synthetic form of the FSH is available and what is their difference with human. So if you look, there is a gonal F, which has the alpha subunits. There is a beta folliotropin, that these two are the majority of the time that we are using. They have, all of them, they have two glycosylation. They are less acidic. And when we compare with the physiologic amount of the FSH, they have different property and half-life. They are less acidic compared to the natural one. Also, not only this, they activate the different pathways. In human FSH, for example, if you consider that menopause compared to follicle. First of all, follicle is 95% pure. Menopause, only 5% pure. And of course, the response of the patients are going to be different because the human one is more acidic. Menopause is more acidic than the recombinant FSH. Now, in normal situation, the pathway that usually will be activated is CAMP pathway. Arrestin, ERK1 and 2 are the secondary pathway. When they use the gonal F with the hormonal and physiological dose, they find out that it doesn't activate the CAMP. They have to increase the dose of medication, the potency of the medication, in order to be able to activate the pathway. And majority of the time, it was preferable to be activated through ERK1 and arrestin 2. So this means that we are activating the different pathways with the different preparation that we have. And this also could have the different effect on response to the treatment in different patients. So what are the problems that we have now with regards to the pharmacogenomic studies in IVF and ovarian stimulation? One, there are not so many JWAS studies. Few JWAS study has been done. Sample number is very low, and it has been calculated that if when we are assuming the common SNP variant to explain about 3.4% change in the response on the treatment in IVF, at least we need 1,000 samples. None of the JWAS study had this number. And we don't have enough number even to do the meta-analysis and find out. So one thing that I'm saying, and I'm calling, is having more JWAS study to have more data with regards to that. Also, the other things that the population heterogeneity in different studies, cost of genetic testing, it's huge. In some way, if the pharmacogenomic going to be a part of the precision medicine and our everyday practice, there should be some way that we could bring the price down. Another part is the difficulty in interpretation of the results because it's early in infancy. There are heterogeneous results that we are seeing. So it means that calling for more research to be done, to have more consistent data to be able to interpret, and also train the physician to know how to interpret these results. The last things is the important things that the nature of the response in COS is really polygenic. At the end, I want to say that the pharmacogenomic studies eventually lead to the era that DNA sequencing of the patients become integral part of the determinant of the drug therapy. So this is going to happen. What we need is to call for more specific, more studies and research, and don't focus only to make the FSH that we are all the time using with the same. We have to understand more physiology of the human to understand what each pathways, when they will be activated, what is the results. And the answer is not increasing the dose. Whenever we increase the dose, it doesn't mean that we are getting the favorable results. And that means that we need really more study in pharmacogenomic aspects and science of the ART.